Hey guys! Today we're going to talk about monoclonal antibodies with the simplified name MABS and how they can be used to fight cancer.
The definition of a monoclonal antibody is an antibody produced from a single clone of cells and consisting of identical antibody molecules.
Let's introduce you first to naked monoclonal antibodies, as these are the most common antibodies used to treat cancer. They are called nakeds because there are no drug or radioactive material attached to them. There are many ways in that these work. Firstly, they can boost a person's immune response against cancer cells by attaching to them and acting as an opsonin so the body's immune system can destroy them. An opsonin is any substance that binds to the surface of a particle to enhance the uptake of the particle by a cell of the immune system. Secondly, monoclonal antibodies can target immune system checkpoints to boost the immune response. These are molecules on certain immune cells that need to be activated or inactivated to begin an immune response. If we trigger these checkpoints, the immune system may be able to know the difference between normal and cancer cells.
Cancer cells often find ways to bypass these checkpoints to avoid being recognised and attacked by the immune system, and therefore if we use drugs that can target these checkpoints, we may be able to stimulate the immune system to kill the cancer cells. Just to help you if you're studying A-level biology, we're going to add this extra bit of information which is linked to our previous statement. T cells are cells involved in the immune response in our body, and if they're not deactivated after responding to an immune attack, they may begin to attack our own cells. Therefore, they have a checkpoint protein on their surface called PD-1. When the protein PD-L1 binds to this checkpoint, the T cell becomes deactivated and doesn't damage any other cells.
And thirdly, naked monoclonal antibodies may work by attaching to and blocking antigens on cancer cells that help them grow and spread. For example, this antibody is the antibody that works against the HER2 protein, which is an antibody found on the surface of breast and stomach cancer cells. When HER2 is activated, it helps these cells grow. This antibody binds to these proteins and stops them from becoming activated.
Let's move on to the next type of monoclonal antibody. It's called a conjugated monoclonal antibody, and they're basically naked monoclonal antibodies that are joined to a chemotherapy drug or radioactive particles. This is useful because the antibody can take the toxic substance directly to the cancer cells, attach to the target complementary antigen on the cancer cell and release it. They are more powerful than naked monoclonal antibodies, but they cause more nasty side effects.
The last type of monoclonal antibody we're going to talk about is a bispecific monoclonal antibody. They are made up of two parts of different monoclonal antibodies, so they can attach to two different antigens at the same time. An example is blinatumomab, which is used to treat some types of acute lymphocytic leukemia. One part of the blinatumomab attaches to the CD19 protein, which is found on some leukemia and lymphoma cells. Another part attaches to CD3, a protein found on immune cells called T cells. By binding to both of these proteins, this drug brings the cancer cells and immune cells together, which is thought to cause the immune system to attack the cancer cells.
Thanks for watching! Bye!